Today we are going to discuss all about microbial genetics. Microorganisms were generally ignored by the early genetics because they are small in size and were thought to lack variable traits and sexual reproduction necessary for a mixing of genes from different organisms. After it was discovered that microorganisms have many different physical and physiological characteristics that are amenable to study, they become objects of great interest to geneticists because of their small size and the fact that they reproduce much more rapidly than larger organisms. Bacteria become important model organisms in genetic analysis, and many discoveries of general interest in genetics arose from their study. Bacterial genetics is the center of cloning technology. So let's discuss now about microbial genetics. Genes are segments of the deoxyribonucleic acid, which is DNA, that contain the code for a specific protein that function in one or more types of cells in the body. Chromosomes are structures within cells that contain a person's genes. Genes are contained in chromosomes, which are in the cell nucleus. Chromosomes has one long double helical strand of DNA. The DNA is tightly wound up and associated with many DNA binding proteins. These proteins are essential for carrying out the two functions of DNA: the replication, transcription, and translation. DNA replication. As you can see, it is the process by which a double-stranded DNA molecule is copied to produce two identical DNA molecules. It is an essential process because whenever a cell divides, the two new daughter cells must contain the same genetic information or DNA as the parent cell. So, why DNA needs to replicate? DNA is the genetic material that defines every cell. Before a cell duplicates and is divided into new daughter cells through either mitosis or meiosis, biomolecules and organelles must be copied to be distributed among the cells. DNA found within the nucleus must be replicated in order to ensure that each new cell receives the correct number of chromosomes. The process of DNA duplication is called DNA replication. Replication follows several steps that involve multiple proteins called replication, enzymes, and RNA. In eukaryotic cells such as animal cells and plant cells, DNA replication occurs in the S phase of interphase. During the cell cycle, the process of DNA replication is vital for cell growth, repair, and reproduction in organisms. Step 1. There are 4 steps in preparation for replication. Step 1 is replication for formation. Before DNA can be replicated, the double-stranded molecules must be unzipped into 2 single strands. DNA has 4 bases called adenine, thymine, cytosine, and guanine that form pairs between the 2 strands. Adenine only pairs with thymine, and cytosine only binds with guanine. Adenine only pairs with thymine, and cytosine only binds with guanine. Step 2. Primer binding. On these steps, the replication begins. The leading strand is the simplest to replicate once the DNA strands have been separated. A short piece of the RNA called the primer binds to the third end of the strand. The primer always bend as the starting point for replication. Primers are generated by the enzyme DNA primase. Step 3. Elongation. Enzyme known as the DNA polymerase are responsible creating the new strand by a process called elongation. During elongation, an enzyme called DNA polymerase adds DNA nucleotides to the three end of the newly synthesized polynucleotide strand. The template strand specifies which of the four DNA nucleotides, adenine, thymine, cytosine, and guanine, is added to each position along the new chain. Termination, it happens when two replication forks meet on the same sketch of DNA, during which the following events occur. Although not necessarily in this order, forks converge until all intervening DNA is unwound. Any remaining gaps are filled and ligated. Castanins are removed and replication proteins are unloaded. DNA transcription. The second part the second part. Transcription is the process in which a gene's DNA sequence is copied or transcribed to make an RNA molecule. RNA polymerase uses one of the DNA strands. The template RNA polymerase uses one of the DNA strands, the template strands, as a template to make a new complementary RNA molecule. Transcription ends in a process called termination. The transcription takes place in three steps: initiation, elongation, and termination. Initiation is the beginning of transcription. It occurs when the enzyme RNA polymerase binds to a region of a gene called promoter. This signals the DNA to unwind so the enzyme can read. The basis in one of the DNA strands, the basis of one of the DNA strands. The enzyme is now ready to make a strand of mRNA with a complementary sequence of bases. Elongation and transcription of DNA is the addition of nucleotides to the mRNA strand. Ribonucleic acid polymerase reads the unwound DNA strand and builds the messenger ribonucleic acid molecule using complementary base pairs. There is a brief time during this process when the newly formed RNA is bound to the unwound DNA. During the process, an adenine in the DNA binds to an uracil in the RNA. Termination and transcription of DNA is the ending of transcription. And occurs when RNA polymerase crosses a stop sequence in the gene. The mRNA strand is completed and it detaches from DNA. Translation in DNA is a process that takes the information passed from DNA as mRNA and turns it into a series of amino acids bound together with peptide bonds. It is essential a translation from one end nucleotide sequence to another code amino acid sequence. Genetic code contained within a messenger RNA mRNA molecule is decoded to produce a specific sequence of amino acids in a polypeptide chain. There are three steps in DNA translation. Those are initiation, elongation, and termination. Almost the same as on transcription. In this stage, in initiation, the ribosome gets together with the mRNA and the first tRNA, so translation can begin. When mRNA o tRNA and amino acid meet up inside the ribosome, each new codon matches with the new tRNA anticodon, bringing in a new amino acid to lengthen the chain. Elongation. It is simply the ribosome traveling down the message, reading codons and bringing in the proper amino cell to translate the message out to protein. The incoming amino acid RNA is brought into the ribosome E site, where it is matched with the codon being presented. Termination and translation of DNA occurs when the ribosome encounters a stop codon, UEG, UEE, or UGE, in the A site. Upon stop codon recognition, the release factor 1 and release factor 2 promote the hydrolysis of the ester bond in the peptidyl tRNA in the P site, leading to the release of the complete protein and termination of the protein synthesis. The only differences of DNA replication is it occurs in preparation for cell division, while transcription happens in preparation for protein translations. DNA replication is important for properly regulating the growth and division of cells. Transcription only occurs when a gene is turned on. Genome. This view like a worm is called genome. It is the complete set of genetic information in an organism. All the genetic that a cell must have to function and reproduce as a cell of a particular species. E. coli has one circular chromosome. This is an E. coli. Has one circular chromosome. That one chromosome is its genome. That one chromosome is approximately 4 million base pairs long. Human genome consists of 23 different autosomal chromosomes and 2 different sex chromosomes. Moreover, each human cell except for sperm cells and egg cells has a pair of each autosome and a pair of sex chromosomes. Therefore, human cells are said to have a diploid genome, while bacteria have a haploid genome. Importance of microbial genetics. Microbes provide relatively simple system for studying genetics phenomenon and those useful to other higher organisms. Understanding genetics of disease-causing microorganisms, especially virus, will be useful to control diseases. That's all.